Szanowni Państwo, witamy na wtorkowej sesji naszego tygodnia Mózgu. Zapraszamy na pierwszą prelekcję. Naszą prelegentką jest profesor Sarah-Jayne Blakemore. Profesor Sarah-Jayne Blakemore jest profesorem psychologii na Uniwersytecie Cambridge w Wielkiej Brytanii oraz liderką grupy zajmującej się rozwojową neuronauką poznawczą. Badania jej zespołu skupiają się na rozwoju poznania społecznego oraz podejmowania decyzji w ludzkim mózgu dorastającej młodzieży i na zdrowiu psychicznym tejże grupy. Jej zespół prowadzi badania behawioralne w szkołach i laboratoriach, a także badania neuroobrazowe z młodzieżą i dorosłymi. Profesor Blakemore jest przewodniczącą Royal Society of Biology Education and Science Policy Committee. Jest także redaktorką w Proceeding of the National Academy of Sciences oraz w komitecie doradczym Lancet Child and Adolescent Health. Za swoje badania otrzymała liczne krajowe i międzynarodowe nagrody, w tym nagrodę doktorancką Brytyjskiego Towarzystwa Psychologicznego czy International Union of Psychological Science Quadrennial Major Advancement in Psychological Science Prize. Jest członkinią Royal Society of Biology, British Academy oraz Amerykańskiego Stowarzyszenia Nauk Psychologicznych. Profesor Sarah-Jayne Blakemore przygotowała dla nas wykład pod tytułem Teenage Brain. Oglądającym przypominam, że możecie Państwo wpisywać pytania do czatu transmisji, przekażemy je naszej prelegentce i odpowiedzi zawrzemy w poście na naszej stronie. Profesor Blakemore także zachęca do kontaktu mailowego. Nie przedłużając, zapraszam. Ladies and gentlemen, welcome to the Tuesday session of our Brain Week. We invite you to the first lecture. Our speaker is Professor Sarah-Jayne Blakemore. Professor Sarah-Jayne Blakemore is Professor of Psychology at the University of Cambridge, UK, and leader of the Developmental Cognitive Neuroscience Group. Her group's research focuses on the development of social cognition and decision-making in the human adolescent brain and adolescent mental health. Her group runs behavioral studies in schools and in the lab as well as neuroimaging studies with adolescents and adults. Professor Blakemore is chair of the Royal Society of Biology Education and Science Policy Committee. She is an editor at Proceeding of the National Academy of Science and on the advisory board of the Lancet Child and Adolescent Health. She has been awarded national and international prizes for her research, including the British Psychological Society Doctoral Award and the International Union of Psychological Science Quadrennial Major Advancement in Psychological Science Prize. She is a Fellow of the Royal Society of Biology, the British Academy, and the American Association of Psychological Science. Professor Sarah-Jayne Blakemore prepared for us a lecture entitled Teenage Brain. I would like to remind the viewers that you can enter your questions in the broadcast chat. We will pass them on to our speaker and we will include the answers in the post on our website. Professor Blakemore also encourages you to contact us by email. Without extending, enjoy listening.
My name is Sarah-Jayne Blakemore. I'm Professor of Psychology and Cognitive Neuroscience at the University of Cambridge, and today I'm going to be talking about our work on the teenage brain. The reason I became interested in the teenage brain is because during my PhD and postdoctoral research, I was investigating schizophrenia, which, as you may know, is a psychiatric condition that's characterized by symptoms like hearing voices inside your head and being paranoid, thinking that other people are out to get you. What interested me was that whenever I asked a patient, when did you start experiencing these symptoms, there wasn't a single exception. The many hundreds of patients I tested all said at some age between 18 and 26 years. So I became interested in why this is. What is it about teenage brain development that is different in teenagers who go on to develop schizophrenia compared to those who don't? And to my surprise, this was in the early 2000s, when I looked into the scientific literature in this area, I was surprised to find that almost nothing was known back then about the development of the human teenage brain. So at that point, I decided to refocus my own research and investigate exactly that. How does the teenage brain develop in humans? Now, it's not just schizophrenia that has its onset in adolescence. In fact, most mental health problems start in adolescence. It's been estimated that 75% of mental illnesses first appear before the age of 24. And you can see that in this graph here. What this graph shows is various different psychological and psychiatric conditions on the y-axis and their average age range in years on the x-axis. And you can see that most of them, their onset clusters around adolescence. Adolescence has lots of different definitions. Recently, we have started to define adolescence as the period of life between 10 and 24 years. It's a unique period of biological, psychological, and social development. When you think about teenagers or adolescents, you might think of kind of stereotypical behaviors like making bad decisions, taking risks, being impulsive, being influenced by friends. And you might also think that these kinds of stereotypical behaviors that we often associate with adolescence are something recent, maybe unique to Western teenagers. In fact, there's a lot of evidence that that's not the case at all. For example, if you look back at historical descriptions of this age group, you find striking similarities with the way we typically describe them today. So going back as far as Socrates, for example, so back almost over 2,000 years ago, Socrates wrote a lot about adolescence and what he called youth. And he was, in the same way that often people are today, very kind of negative and stereotyping about this age group. He said things like they have bad manners, contempt for authority, they show disrespect for elders and love chatter in the place of exercise. Aristotle too wrote a lot about youth. This is just a couple of sentences taken from large passages that he wrote about youth, saying things like they're passionate, irascible, and apt to be carried away by their impulses. This is the age when people are most devoted to their friends, and that's a concept that I'm going to come back to throughout this talk. So these kinds of negative stereotypes about adolescent typical behaviour are nothing new. They've been around for millennia. Shakespeare as well, about 400 years ago, wrote a lot about teenagers. This is an example from The Winter's Tale where he says, I would there were no age between 10 and 3 and 20, or that youth would sleep out the rest. For there is nothing in the between but getting wenches with child, wronging the ancient tree, stealing, fighting. Again, very negative stereotypes about this age group. I mean, why these negative stereotypes about adolescents are socially acceptable today and seemingly have always been is another question. I don't know, and it's something that I think about and worry about, because it seems strange to me that it's socially acceptable to malign and mock this sector of society when it's not really acceptable to do it to any other sector of society. What about across cultures? So typical teenage behaviour, these kind of stereotypes of behaviour that we have about teenagers and adolescents, so things like risk-taking and impulsivity and peer influence, are they confined to Western adolescents? Actually, there's evidence that they're not. So not a huge amount is known about adolescent development in terms of cognition and brain development across cultures. But there was a study carried out by Larry Steinberg and his colleagues a few years ago in young people in 11 different cultures. So these are very different cultures in different countries around the world. And he looked at various different behaviours and how they develop across cultures, how they develop across adolescence in different cultures. And what he found was some, again, striking similarities between cultures in developmental trajectories of behaviours such as sensation seeking. So sensation seeking is defined as the desire to seek novelty and to take risks. And this is measured by a composite of experimental tasks and questionnaires. And what you can see here is how sensation seeking averaged across 11 different countries, which is this dark line here, how it develops across age from 10 to 30 years. And although there was some variation across cultures, what I think is probably most striking is that the trajectory is similar. generally across these different countries, in that sensation seeking and risk taking increases during the teenage years. It's highest in the late teenage years, and then it decreases again quite substantially during the 20s. A different behavior, self-regulation, that is the ability to regulate your actions and to plan your actions and to inhibit yourself taking risks, again showed similar trajectories across these 11 different cultures, improving gradually during the teenage years and then stabilizing in the early to mid-20s. So across cultures, certain types of adolescent typical behaviours seem to be developing in similar ways. Now, one of those behaviours, as I mentioned, is risk-taking. There is a lot of research on risk-taking around the world in adolescence, and I think that's partly because we worry about the risks that adolescents typically take. This is because a lot of research has shown that risk-taking can result in accidents and even mortality. So, for example, it was found that in Western cultures, the leading cause of mortality in people aged between 10 and 24 years is accidents, mostly caused by risk-taking. So in a way, this is interesting because that's not true in any other age range. This seems to be specific to this adolescent age. And it's also interesting because in theory, these kinds of this mortality, which results from accidents, is preventable. And so I think that's part of the reason why a lot of research and policy decisions are focused on adolescent risk-taking. But actually, adolescent risk-taking isn't all bad, and in this stereotype that all adolescents take risks isn't, of course, true. There's lots of individual differences. Some adolescents are risk-averse, some adults are very risk-taking. Often risks are a good thing. We have to learn by risk-taking through trial and error, through exploration of our environments. And finally, risk-taking in adolescence tends to be very specific to the context, in particular the social context. We know that adolescents are much more likely to take risks when they're with their friends than when they're on their own. Now, lots of research has shown this. For example, research in labs, but also from real life data, for example, data from car insurance companies and from epidemiological studies. And in this case, this graph I'm just about to show from the American Automobile Foundation. So what this shows is the percentage increase in the risk of a fatal accident, so death risk, when young drivers under the age of 21 are driving with passengers versus on their own. So what you can see here is with one passenger, there's around a 40% increase in having a fatal accident compared with when young drivers are driving by themselves. And with each additional passenger, there's an exponential increase in the risk of a fatal accident. What this suggests is that there's something about having passengers in the car which increases the risk of a fatal accident when young people are driving. And this phenomenon has now been taken into account in policy decisions. For example, in Canada and Australia, there are rules about young people not being able to drive with passengers who are not family members before a couple of years after they've passed their test. The precise rules depend on the country. And some car insurance companies also offer reduced premiums for young people as long as they don't carry passengers in the car with them. The peer influence on risk-taking has also been shown in lab-based studies. For example, this study, which was carried out by Larry Steinberg and his colleagues, asked participants to come into the lab and carry out a driving game where they had to drive around a circuit, like a kind of video arcade driving game. Now, they measured the number of risks participants took, for example, the number of red lights they drove through and crashes they subsequently had, and they tested different age groups. This is the number of crashes, so the number of risks people took when they were on their own. Adolescents aged 13 to 16 in blue, young adults aged 17 to 24 in yellow, and adults aged 25 and over in red. And what you can see is that when they're on their own, each group takes around the same number of risks and has the same number of crashes. However, in a second condition, the experimenters asked the participants to bring a couple of friends with them, and those friends stood behind the participants as they carried out exactly the same driving game. And in that social condition, the number of risks, the number of crashes adolescents had was about 3 times as many as when they're on their own, and about twice as many in the young adult group. And having friends with them had no effect on the adult group's risk-taking, shown in red. So again, this lab-based study shows that peers have a really significant effect on decision-making and risk-taking in adolescence, but not so in adulthood. Now, what's interesting is that this peer influence on risk-taking doesn't seem to be unique to humans. There's a lot of evidence that adolescent animals, so all species of animals, undergo a period of life between puberty and becoming fully sexually mature adults. You can call that adolescence. And in adolescence, animals have been shown to, for example, mice and rats, they're often studied. They go through about 35 days of adolescence. And in that 35 days, they have been shown to take more risks, to explore their environment more, and even to show really big changes in social behavior. For example, one study showed that adolescent mice drink more alcohol when they're with other mice. And that's not the case for adult mice. So you can see that here. This is the time spent drinking alcohol when they're on their own. The adolescent mice in blue, the adult mice in red. Whereas when they're put with their cage mates, the adolescent mice drink significantly more alcohol than when they're on their own. But this social effect isn't present for adult mice. So it seems that the social effect on behavior is not unique to human adolescents, but is also present in other species of adolescents. So in my lab, we're really interested in this social influence on risk perception. And one study that we carried out a few years ago, which was led by my former postdoc, Dr. Lisa Knoll, we tested 563 participants, about half of whom were female, aged between 8 and 59 years. And in this study, they had to do a very simple paradigm where they looked at a computer screen, a laptop, and they were presented with various different moderately risky scenarios. So for example, crossing a street on a red light is an example of a scenario. There were many others, like driving without a seatbelt or cycling without a helmet. And after each scenario, participants are asked to rate the riskiness of this particular situation on an analog scale using a computer mouse. And then they're told, all the teenagers who've taken part so far in this experiment, this is their average risk rating for that same scenario. In a completely different condition, the participants are told this is the average risk rating of adults who've taken part in this experiment so far. Now, the important thing here is that this provided rating isn't really from other teenagers or adults. It's randomly generated by the computer. But participants believe that they're seeing the risk ratings averaged from other teenagers or adults. And then we asked them to rate the same scenario again. And what we're really interested in is whether participants change their rating from time 1 to time 2 in the direction of the provided rating. That gives us an indication of how socially influenced they are. And what we're particularly interested in, and this is what I'm going to show you today, is whether people are more socially influenced by adults' risk ratings than by teenagers' risk ratings. That's what our hypothesis was. We thought that people would be socially influenced because decades of social psychology research has shown that social influence is very strong in all of us. But we thought that if people are going to be influenced by others, they're more likely to fall in line with the risk ratings of adults than teenagers. So I'm going to show you that on the next graph. Okay, what you see here is the 5 participant groups. These are the groups that we tested down here on the x-axis. So children aged 8 to 11, this is their age range in years, young adolescents, mid-adolescents, young adults, and adults. If they were more influenced by adults than by teenagers, the bar will be below the 0 line. And that was the case for the children and the 2 adult groups in our study. So they conformed to other adults more than to teenagers. In contrast, the mid-adolescents showed no difference. In other words, they were equally influenced by both adults and teenagers. And in contrast again, the young adolescents aged 12 to 14 showed exactly the opposite pattern of behaviour. They were more influenced by teenagers than by adults. So what this suggests is that young adolescents aged 12 to 14, their risk perception is more influenced by people their own age than it is by adults. Now, this is a finding that we replicated. So we showed it in this original study of 563 participants. That's the data you're seeing here. And we replicated it in a second study of 590 completely different group of participants. So it seems that this effect here is robust and can be replicated. And what it suggests is that young adolescents are really influenced by each other. Now, this has implications for all sorts of things, but including public health. So now most public health advertising focuses on the health or legal risks of certain behaviours, like, for example, I don't know, anti-smoking or anti-experimentation with drugs or anti-bullying or health eating campaigns. They focus on the negative health consequences of these behaviours. But actually what this suggests is that maybe what would work more if you're targeting young people is focusing on what their peers think and the social norms of their age group. And indeed, this has been shown in a study carried out a few years ago, looking at peer-led anti-bullying interventions in schools. So this is a study that was carried out in New Jersey in the USA in 56 middle schools. So this is children aged between 11 and 15 years. So these 56 middle schools were divided randomly into two groups. In one group, the anti-bullying and social exclusion campaigns were led by the teachers, as they usually are. And in the other group of schools, the anti-bullying campaigns were led by the students themselves. So a group of students was selected, they were educated about the harmful effects of bullying and social exclusion, and then they were incentivized to run their own campaigns. So they ran things like debating campaigns, wristband campaigns, poster campaigns, all sorts of things. And what they found over the ensuing year is that bullying incidents and other social exclusion incidents in the schools were reduced by about a quarter over the ensuing year in the schools in which the interventions were led by the students, compared with the schools in which the anti-bullying campaigns and interventions were led by teachers. And the effect was stronger when the students who were leading the anti-bullying campaigns were the more highly connected students in the year group. So this has not only been shown for anti-bullying, but also anti-smoking, and it suggests that empowering young people to devise and run their own campaigns can have really positive effects on behaviour. So one of the questions we have investigated is why adolescents are so susceptible to peer influence. And one of our hypotheses was because adolescents might be particularly sensitive to peer rejection. So they might have a kind of increased need to affiliate with their peer group and go along with their friends. One way of testing this was to look at the effects of being socially excluded on mood and anxiety. And we did this, this was a project that was carried out by my former PhD student, Kat Sebastian. We did this using Cyberball, which is an online ball throwing game. I'll just show you the game. So here you are represented by this hand here, and you're told you're going to play a ball throwing game over the internet with two other players. And either they can include you in their game or they might not. But what you're not told is that, in fact, there are no other players and we control whether you're included or excluded in the game. I'll show you an example of an exclusion condition. So in this condition, you're throwing the ball and you decide who to throw it back to, and they could decide to throw it back to you, but in this case they don't. And for about 90 seconds, they just continue to play the game with each other and they leave you out. And that reduces your mood and increases your anxiety. It makes you feel sad and anxious. That's been known for many decades. This cyberball game was originally designed by Kip Williams and his colleagues, and they and many others have shown that in adults, social exclusion, just in this very brief, online, simplistic form, results in lowered mood and increased anxiety. So we wanted to know whether that was the case for adolescents. And in this study, we used cyberball with young adolescents, mid-adolescents, and adults. What you're seeing here is the results for mood as measured by a standard mood questionnaire in our adult group. So these are adults aged 22 to 47. At baseline, yellow here, that's their mood at baseline before they've played any game. The green bar represents their mood after being included in cyberball. By the way, that's not a significant increase in mood after its inclusion, implying that we expect to be included. It doesn't particularly increase our mood, but there is a significant drop in mood after being excluded on this game. So the young adolescents and the mid-adolescents showed the same pattern of results in terms of mood. The difference was that their drop in mood after being excluded in cyberball was significantly greater than the drop in mood after exclusion in adults. And what that suggests is that adolescents are hypersensitive to social exclusion. Now, if adolescents are hypersensitive to social exclusion, then this might lead them to really want to avoid being socially excluded. Of course, none of us wants to be socially excluded, but perhaps the pain of social exclusion or social rejection is even greater if you're an adolescent. And if that's the case, that might have an effect on decisions you make, your behavior, your actions. So for example, if you think about a typical teenage girl who's out at the weekend and all her friends are smoking, for her, it might be wise in a way to go along with this health risky behavior, because it will mean that she probably avoids being socially excluded. So I suppose a way of saying this is, for her, what is the bigger risk? Saying yes to a cigarette when she knows that cigarettes can lead to health risks, or saying no and potentially being ostracized by her peers? Well, we would argue that for an adolescent, avoiding social risk, in other words, avoiding the risk of being socially excluded, might weigh in more heavily than avoiding other kinds of health and legal risks. So avoiding social risk might matter more to adolescents than avoiding other types of risks. And this explanation might shed light on why educated, intelligent adolescents might sometimes make decisions that to us adults seem irrational, but perhaps they're not so irrational when you think about the need to be included by the peer group is heightened, and also probably for adaptive reasons. After all, adolescence is the period of life when we need to eventually end up becoming independent adults. And part of that is affiliating with peers and figuring out where you are in the social hierarchy. Peers and friends really matter to young people. That's not to condone these kinds of decisions. Of course, it would be better if they didn't make these kinds of health risky decisions, but it sheds those decisions in a more rational light. So in the rest of this talk, I'm going to turn to how the brain develops during adolescence. Now, when I was an undergraduate about 25 years ago, I was taught that the human brain stops developing in childhood. I remember that, and I also kept my undergraduate textbooks, and that's exactly what they say. So this is an example of a paragraph from one of my undergraduate neuroscience textbooks, and it's talking about myelination, which is a really important neurodevelopmental mechanism. It says myelination begins usually late in embryonic life or early in postnatal life, etc., etc. It continues for considerable periods of time into childhood in the case of humans. Now, what's really interesting is, as this implies, most research back then had been done on animals, and we just didn't really know how the brain develops in humans. And it was assumed that the human brain stops developing in childhood. And that's because back then we did not have the technology to look inside the living human brain to study how it changes with age. We now are able to do that using magnetic resonance imaging, MRI scanning. MRI scanning is very common in hospitals and for research purposes. In terms of what it can show you in the brain, we use MRI to measure the structure of the human brain. That's what you can see here. This is one of my colleagues. This is her MRI scan. Structural MRI scans allow you to measure the amount of grey matter in the brain. Grey matter is mostly found in the cortex, the surface area of the brain, and grey matter contains cell bodies, so neuronal cell bodies, and synapses, the connections between cells, amongst other neural matter. You can also measure white matter in structural MRI scans. White matter are the long fibres, which are called axons, that connect up different regions of the brain, allowing them to communicate with each other. So you can record brain images in the living human brain at all ages to track changes in the structure of the brain across the lifespan. You can also look at functional MRI, that is, where in the brain is active when participants lie inside the scanner and do a particular cognitive or perceptual task. Now, over the last 2 decades, many researchers around the world have studied the development of the living human brain using MRI scans. And those studies point to a very robust and replicated finding, which is that, contrary to what I was taught as an undergraduate, the human brain in fact undergoes very substantial and protracted development right throughout childhood, but also continuing throughout adolescence and into early adulthood. Now I'm going to show you an example of how the brain changes, and this example comes from a study that was led by my former PhD student Kate Mills and our colleague Christian Tamlers in Oslo. They looked at structural brain development in 4 different cohorts, so 4 different groups of child and adolescent participants. These 4 cohorts were from 4 completely different places: NIH in Pittsburgh in the USA, and Leiden in the Netherlands, Oslo in Norway. So in total there were 391 participants across all those different places. And each one of those participants joined the study when they were a child or an adolescent, and then they were scanned repeatedly as they grew up. In total, there were 852 MRI scans that were analysed in this data set. I'm going to show you a couple of the key findings from this data set, although there are many more, and you can look at them in the papers that are cited on the slides if you're interested. So one of the main findings, which has been now found in many different MRI studies of development, is that cortical grey matter changes across age, and that's what you're looking at here. So cortical grey matter, that's the grey matter that's found in the cortex, across the whole cortex, is plotted here. Its volume is plotted against age and years from 5 to 30. The colours correspond to the 4 different cohorts. Now, there are 2 things that you should notice here. First of all, how remarkably similar the developmental trajectory of grey matter volume is in the 4 different cohorts, even though they're completely independent groups of people growing up, living in different countries in some cases. And the second thing to notice is the shape of the developmental change. This is a cubic trajectory where grey matter is highest in late childhood, so grey matter increases in childhood, you can't see that here. It's highest in late childhood, and then it undergoes this very substantial decline across the whole of adolescence and only starts to stabilise in the mid-20s. In fact, grey matter volume decreases by about 1.5% annually during adolescence. I'm going to show you a video of that. Now, this video actually comes from an earlier study, which includes the NIMH sample of people. What it shows is how grey matter changes across the brain, across age, between the ages of 4 and 21 in this case. So grey matter here, this is the brain, the back of the brain here, the front of the brain here, and the brain is colour coordinated. So the more grey matter it contains, the more red and yellow it is, and as grey matter is lost, it becomes more blue. So this is age 4, and gradually with age, you can see that grey matter undergoes substantial decline. I'm going to play that again because the second thing to notice is that it's not uniform. Different brain regions lose grey matter at different rates. And generally these studies have found that the prefrontal cortex and other anterior regions of the brain undergo the most protracted development, as opposed to the back of the brain, which contains the visual cortex, and other sensory regions like the auditory cortex and the motor cortex, which controls movement, develop earlier. but the prefrontal cortex and other frontal regions develop latest. And they are involved in all sorts of high-level cognitive processes like decision-making and planning and inhibiting inappropriate actions, self-awareness, social awareness. Now, grey matter declines dramatically during adolescence. That might sound like a negative thing, like neuroatrophy or neurodegeneration. In fact, we don't think it's that at all. We think it represents really important neurodevelopmental processes. And that will be explained if I tell you that the second finding from these developmental MRI studies is that at the same time that grey matter is decreasing, cerebral white matter is increasing, and that's what you're looking at here. So this is the volume of white matter in the brain, and again plotted against age in the 4 different cohorts. Again, the 4 different cohorts show a very similar trajectory of white matter, which is a linear increase. White matter volume increases by about 1% each year during adolescence. Now, the question is why? Why does grey matter volume decrease and white matter volume increase during adolescence? MRI scans, although they give us these beautiful pictures of the living human brain, and they've completely changed the way we think about how the human brain develops, they don't have yet the resolution to tell us very much at all about the brain at the level of the cell or the synapse. So we can only make educated guesses about what's happening at the level of the cell and the synapse to underlie these changes in grey matter and white matter based on what we know from animal research and also research involving post-mortem human brain tissue. And from that research, we know that there are at least 3 really important neurodevelopmental mechanisms that are occurring in the brain during adolescence. The first is myelination and also axonal growth. Now, to explain what these are, I need to show you a diagram of a neuron. So here is a neuron. This is the cell body. It generates electrical impulses which pass down this axon, this fibre, and are then communicated with other neurons in the surrounding area via this synapse here. Now, in development, this axon, a few things change to the axon. Firstly, it grows in diameter. Secondly, it becomes coated with what's called a myelin sheath. It becomes myelinated. And those 2 processes, both the increase in diameter and the myelination, effectively increase the speed of transmission of electrical signals moving along the axon. And we know that both these processes, axonal increases in diameter and also myelination, are happening, well, from about the 2nd trimester actually in pregnancy in humans, but they continue throughout childhood and throughout adolescence and even into early adulthood in humans. So this is a little cartoon showing the difference between a myelinated and an unmyelinated axon in terms of the speed of signal transmission down the axon. The top one is myelinated, and you can see that the signal is passing much faster down the axon than the bottom neuron, which has an unmyelinated axon. That's just a cartoon, but there's a huge amount of research corroborating this finding that myelin increases speed of transmission. So myelination and axonal growth will correspond in MRI scans to an increase in white matter and a concomitant decrease in grey matter. And that is because after the age of about 8 or 9, the human brain doesn't really grow in volume at all. So the brain stays the same size, but its internal composition changes. It becomes more white as axons are coated in myelin, which is white, and they grow in diameter. And therefore, there's a concomitant decrease in grey matter, as grey matter essentially becomes white matter. The third developmental process that's happening in adolescence is synaptic pruning. So synapses, these connections between cells, they are overproduced across the brain in early development. The brain produces an excess number of synapses. Those excess synapses are not specialized for function, and the excess synapses need to be eliminated. And that happens through a process called synaptic pruning. Now, we know a couple of things about synaptic pruning in this context. First of all, that in some regions of the human brain, like the prefrontal cortex, synaptic pruning continues right throughout adolescence and even into the 20s and 30s. We know that from post-mortem human brain tissue studies. We also know that an interesting fact about synaptic pruning is that it's partly dependent on the environment that the animal or human is growing up in that synapses that are being used in that particular environment will remain and grow stronger, and synapses that are not being used in that particular environment are the ones that become eliminated and pruned away. And in that way, the environment can help to shape the development of the human brain. We also know that synapses are found in grey matter, and so as synaptic pruning happens, this might correspond to a decrease in grey matter as viewed by an MRI scan. However, synapses are tiny, and this probably only accounts for a very small decrease in grey matter. Now, my final slide turns to individual differences. So far in this talk, I have really been talking about average teenagers, but of course there is no average teenager. Teenagers are all different, and so are their brains. But averages have been focused on, and until recently we have downplayed or even ignored individual differences in our science. Individual differences are often kind of either just reduced by looking at homogeneous samples or reduced by statistically in our analysis. But actually individual differences are really, really important. So I'm going to show you an example of this. This is the grey matter graph that I showed you earlier from the 4 cohort study. And what I showed you earlier, you might remember, are these lines of best fit showing grey matter change across age. But what you can see underneath these lines is basically what they are trying to model, and that is the individual data. So each of these points is an individual child, and how that individual child's grey matter changes across a few years of its life. And what you can see is there are enormous individual variation, both in terms of the intercept. So some children, say age, I don't know, 12, have brains containing this much grey matter, while others have brains containing this much grey matter. And also in intercept. So some people's brains undergo a steep decline in grey matter in early adolescence, which then levels off in later adolescence. And some individuals show the opposite trajectory, where nothing much changes in early adolescence, and then there's a steep decline in later adolescence. So we're now becoming, as a field, really interested in what this means. First of all, what is it about someone's genetics and their environment that determines how their individual brain develops? And secondly, what does it mean in terms of outcomes? So one hypothesis, for example, going right back to the beginning of my talk, which was where I mentioned mental illness, perhaps one hypothesis is that perhaps the specific trajectory of brain development is a kind of indication or even a risk factor for developing different types of mental health problem. And that's a big area of research right now. Okay, so just to summarize my talk, I have told you that social risk avoidance is an important determinant of adolescent behavior. The brain undergoes very substantial and protracted development right throughout adolescence, and adolescence as such might be a sensitive period of brain development. Remember, I told you that these very important neurodevelopmental mechanisms are happening in adolescence, and they might render adolescence a sensitive period of brain development where it's particularly plastic and particularly sensitive to environmental experience and environmental input. And this would confer both vulnerability to things like dangerous risk-taking and mental health problems, but also opportunity. If the brain is undergoing a period of heightened plasticity, then this is a really excellent opportunity for things like learning and interventions and rehabilitation. So thank you for listening to this talk, and I also wanted to thank my research group. These are the members of my research group who at some point have been involved in the research I've talked about today, and also my funding. Thank you.
Dziękujemy profesor Blakemore za przygotowanie tak interesującego wykładu oraz dziękujemy naszej publiczności za wysłuchanie tego też wykładu. Zapraszamy Państwa na kolejny wykład, który odbędzie się już o 16:00 i przedstawi go pan profesor Lars Kai Hansen. Thank you, Professor Blakemore, for such an interesting lecture, and thank you for the audience for listening this lecture. We invite you to the second lecture today, which is prepared by Professor Lars Kai Hansen, at 4 p.m. Thank you and see you there.